This is Lecture 2 in Module 3 titled Pathogens and Disease. Infectious diseases are caused by pathogens. A pathogen is any microbe, live or dead, that causes a disease. Not all microbes are pathogens, and they are essential for life on Earth. A good example are the microbes you learned about in Module 2 that are found in the ruminants of animals that digest fiber. Without microbial decomposers, the planet would be covered in dead organic matter. The most common pathogens that can cause disease are viruses, bacteria, fungi, prions, and protozoans. We will cover both protozoans in a later lecture on parasites. Viruses are considered non-living and are composed of two parts, a protein coat called a capsid and either a strand of DNA or RNA. This genetic material takes over a cell and causes it to reproduce or replicate more virus. Viruses attack specific tissues, commonly the skin, blood, liver, uterus, fetus, brain, lungs, and intestines. A decrease in lymphocytes and a complete blood count, CBC, often indicates a viral infection. There are few treatments for a viral infection. Antibiotics do not work on viral infections, so providing support while the infection runs its course is usually the best course of action. Bacteria are organisms that lack a cell nucleus or any membrane encased organelle and are called prokaryotes. Many are essential for life as we know it. Some are major decomposers, photosynthesizers, and are not pathogenic. Bacteria can reproduce rapidly, and pathogenic varieties usually form colonies. They come in various shapes: cocci, which are spheres, bacilli, which are rods, or spirella, which are spirals. Pathogenic varieties attack cells or produce toxins. Symptoms of bacterial infections depend upon the type of bacteria. Samples of the infectious bacteria can be cultured to identify strains. Bacterial infections can be treated with antibiotics, but unfortunately, bacteria quickly become resistant to many of these antibiotics. We must look at the size, shape, and color of a cultured sample, the metabolic attributes or DNA, to identify bacteria. It's very important we are able to identify the type of bacteria so your veterinarian can recommend the type of antibiotic to treat the bacterial infection. Usually the first test done is to stain the colony to distinguish between a gram-positive or gram-negative bacteria. A gram-positive bacteria have cell walls containing peptidoglycan. Examples are Staphylococcus, Streptococcus, and Clostridium. Gram-negative bacteria have cell walls composed of lipopolysaccharides. Gram-negative bacteria are harder to control with antibiotics, and good examples are E. coli and Salmonella. Antibiotics are used to treat bacterial infections. An antibiotic is any substance produced by a microorganism which kills or inhibits growth of another microorganism. Antibiotics generally do not harm the host. By strict definition, the word antibiotic refers to substances produced by microorganisms that act against another microorganism. Thus, antibiotics do not include antimicrobial substances that are synthetic or semi-synthetic, or those that come from plants or animals. Review the slide for good examples of those antimicrobial substances. In contrast, the term antimicrobial includes all agents that act against all types of microorganisms, bacteria, viruses, fungi, and protozoans. Antibiotics must target a pathogenic bacteria. Antibiotics are classified according to the range of bacterial species affected by the product. Broad-spectrum antibiotics are effective against both gram-positive and gram-negative species. Narrow-spectrum antibiotics are only effective against particular species of microorganisms. Please review the slide for examples of both broad-spectrum and narrow-spectrum antibacterials. Fungi are single or multicellular decomposers that sometimes attack live tissue before we're dead. Symptoms include skin lesions, hair loss, respiratory or digestive disorders. Fungal diseases can be treated with antifungal drugs, topical medications, or shampoos. Prions are an abnormally folded protein. They are not alive, which cause the formation of other abnormal protein molecules in the surrounding tissue, especially the brain. Abnormal prions in the brain cause mad cow disease in bovids, chronic wasting disease in cervids, and Jakob-Creutzfeldt or possibly Alzheimer's disease in humans. Let's talk about disease or pathogen transmission. Disease can be caused by transmission of pathogens by direct contact, that is animal-to-animal contact, either through tissue, bodily fluids, placenta, or through sexual contact. We can also get disease transmission through indirect contact. Indirect contacts can occur through feeders, foods, air, sharing of food, the water's soil, or there are animal vectors such as insects, or in our case, birds, pigeons, down at the reindeer facility. In disease transmission, the portals of entry are varied as well. The point of entry can be through the mucous membranes of the respiratory or gastrointestinal tract. We can, our animals can catch a disease and pick up a pathogen from bites, cuts, or injections. You, the animal owner, can prevent or treat disease caused by pathogens by using antibiotics for bacterial infections or vaccinations, which are the bacterial or viral infectious diseases. A vaccination prepares the immune system by forming antibodies that attack the bacteria or virus. Different infectious diseases require different approaches for prevention and control. But for any disease, there are three very important key steps. First, we must break the cycle of transmission. Next, we must kill the infectious agent. And thirdly, we must increase host resistance. Okay, how do we do that? First, we can eliminate animal-to-animal contact. We can quarantine sick animals or cull them if necessary. We can have foot baths for human traffic going from a quarantine to a safe area. And also, you should consider changing outer clothing. We don't want to pass on the pathogen from sick to healthy animals by indirect contact. We can also try to disinfect the environment, clean the feeders, clean the waters. We can line the pastures to kill the pathogen. We can also vaccinate the animal. Vaccination is the administration of a material that stimulates adaptive immunity to a disease. Certain principles are common to all types of vaccination. So when you go to vaccinate your animals, you need to remember some of these principles, or all of these principles. First, newborn animals can be passively protected by maternal antibodies. Neonates, lactating mothers who have just given birth produce a substance called colostrum. And the neonates can acquire immunity from nursing the colostrum because there are antibodies within this colostrum. If stimulation of immunity is necessary before the mother gives birth, the mother can be vaccinated during late pregnancy, and some of these antibodies are picked up by the fetus. Because the exact time of loss of maternal immunity cannot be predicted, young animals are often vaccinated at least twice to ensure successful immunization. The interval between vaccine doses depends on an animal's immunological memory and the vaccine. The duration of this memory depends upon factors such as nature of the antigen, the use of a live or dead organism in manufacturing the vaccine, and the route of administration. Some vaccines may induce immunity that persists for an animal's lifetime, but not always. Other vaccines may require boosting every couple of years, or in some cases, every 6 months. Current research is now developing or modifying plants to contain vaccines. We're actually injecting a vaccine into the plant. These plant-based edible vaccines would be safer, cheaper, and could be grown or freeze-dried and shipped anywhere. The vaccines delivered in this manner have a two-way immune response. You both get a systemic response, but you also get some mucosal immunity. And this mucosal immunity fights infections where the pathogen first enters the body, the mucous membranes of the nose, mouth, lungs, gut, and genitals.